So what we started with this year is offering 6 basic training modules. And these 6 basic training modules are Introduction to Bioinformatics, Genomics, Transcriptomics, Metagenomics, Epigenomics, and Data Science. So these are offered as year-long curricula that you can organize into an asynchronous or a live type course. Our team supports it and actually runs a number of these programs throughout the year. And Dr. Mazumdar will be speaking about some of these programs when I'm done with my part of the presentation. In addition to that, we also provide specialization tracks, and these include oncology, infectious diseases, neuroscience, and agriculture. And agriculture includes also some elements of biotechnology. So what we are trying to do is develop exemplar projects with curated data sets and an explanation of methods that are sourced from impactful publications in that domain to illustrate how some of those basic data types as well as methods could be applied to solve real research challenges. Now, let me briefly explain what is the difference between what I am describing as a curriculum or as a content database and some of the practical resources that are included in this program and in this training. So the OmicsLogic program resources are really designed around training materials, computational tools that are easy to use that biologists can use, interactive coding exercises that could be, first of all, checked for syntax, and second of all, checked for the correct results. And then finally, an introduction to a coding environment where you can take some of the scripts that you've learned how to write and then execute them on a big data infrastructure. So let me briefly introduce some of these resources in greater detail. One resource that is very critical for a biologist to get started is no-code bioinformatics. And that means that you can use bioinformatics without writing a single line of code. And for that purpose, we have a big data analysis platform that is not unlike Galaxy and some of the other tools that exist out there, but it is tightly integrated with the curriculum that we have designed for different types of data. And I'll briefly show this platform today so that you can all get a sense of what it really looks like and how it works. The second part of this tutorial approach is incorporating data science, and that means coding exercises that check your ability to write correct syntax without errors and provide you with specific results to the coding challenges so that you can understand how to get the desired outcome in your code following a tutorial. And then finally, we also are building a network of mentors that can help you take those skills further. And so essentially by working on exemplar projects and trying to identify research questions that could be answered with data analysis skills, we can then eventually match you to a research experience in which you can try to understand a topic with the help of a mentor that will eliminate some of the dead ends and guide you to identify a low-hanging fruit in terms of being able to answer a research question as an experience for you to gain computational biology research skills. Let me briefly show you the resource that I mentioned before. So right here, if you go under courses on the portal, and by now I hope that most of you have already created an account. I saw that some people joined maybe a little bit late, so I'll just remind everyone. This is a portal that we are speaking about called Omics Logic. So you can log in by following the link learn.omicslogic.com. Login is free. You can gain access to most of the resources for free. And if you are a part of one of our programs through the NAIPI, the National Association of Idea Principal Investigators, you can gain access to all of the resources that I'm mentioning today for free using the NAIPI contract that our company has. So here you will find different types of courses, materials, and projects. Right here, you will find different tags that you can try. And below are collections that you can find focused on different types of data, different types of skills. And one of those skills is the Bioinfo platform that really establishes the logic of analysis applicable to big data. So what is this platform? This platform, and by the way, anyone that wants a demo account on this platform, after you have registered on the Learn portal and you have received your password in your email, after 24 hours, you will gain automatic access to this platform just to explore and see what it can do. So this platform is organized according to several different sections. You can see here transcriptomics, genomics, epigenomics, and different methods of analysis listed on this side. There's also sections related to mass spectroscopy and structural biology. There are specific sections in application to cancer biology, single cell analysis, virology, as well as data integration and modeling. So as you look through these different sections, you'll find that some of them have a drop-down button. And this drop-down button gives you access to a demo pipeline builder that you can try without having to upload any data and without having to perform the actual analysis. So here, for example, you will find that informative pop-ups that are associated with each step of the analysis will guide you to build meaningful combinations of scripts and algorithms that will produce results that you can use. So for example, in this transcriptomics section, you can see that I can perform mapping using bowtie and then visualize the SAM file of the mapping that I performed. And in addition to this visualization, I can also find resources on the Learn portal that will allow me to understand what exactly I'm trying to do. So this is just an example. Again, you can see that in many sections, there's a drop-down that you can find different types of methods of analysis and different types of pipelines that you can perform. So you can try and use that demo account once you have created a login. So again, that could be found using coursework on Bioinfo, and you can see here a collection of courses that you can explore and really learn how to perform analysis of metagenomic, genomic, transcriptomic, and other types of data. Now, to illustrate how these kinds of methods are used in a training experience, I want to briefly talk about several different types of programs that we run throughout the year. And then I will pass it on to Dr. Mazumdar to speak about specific programs that we have running right now, starting in the end of January. So a good example of a program that we regularly run is a program on transcriptomic data analysis. And so we call this bulk and single cell transcriptomics. The program is designed to introduce the omics technologies, including how the library preparation happens, how the actual sequencing happens, how does the understanding of the methods of data preparation impact our analysis approaches to that data, and ultimately how to then use some of this methodology to actually be able to perform large-scale analysis using these kinds of methods. So again, starting from the Bioinfo platform that you just saw, we will go through some example pipelines that students build. They build those pipelines to understand the inputs and the outputs behind each step, as well as the logic of analysis. How can they apply the right combination of scripts and methods to achieve the desired results? And all of it is very intuitive, right? As you can see, there's no coding involved. In addition to that, the platform is color-coded, and this color coding follows a logic that we explain in detail throughout the training program itself. To apply those methods, we then introduce several different projects. For example, cancer macrophages, modeling cancer precision medicine, PDX tumor in the microenvironment, but also elements of infectious diseases like malaria infection, or even applications in biotechnology and agriculture like potato drought resistance for modified crops. So that gives you an understanding of the specific skills or specific methods that need to be used, and the specific applications where you can actually try and run that kind of analysis and see what are the results, what type of insights you can generate analyzing such data. In addition to that, we then go on to one of the maybe most exciting areas of transcriptomic data analysis, which is single cell RNA-seq analysis. And we also have a very basic processing pipeline, as well as a downstream analysis pipeline for Seurat and Monocle, that we go from the logic and then apply all of the logic in terms of code that could be run on Google Cloud platform using Google Colab. So what happens is that you understand the logic, the need, and the reason for specific methods, and then you can apply the specific portions of that code to try and modify and get the types of figures that you might need for a publication. Moreover, you will then see how the exact same approaches are used in high impact publications that are really transforming our understanding of different domains, from cancer to neuroscience, using such techniques. So again, the objective in such a program, in such a basic program, is to introduce the specific type of data, the methods that are typically used to analyze such data, and then showcase some of the applications of that data analysis, including multi-omics integration. Another type of a program that I want to briefly speak about is bioinformatics for infectious diseases. So one of the things that really happened over the past couple of years is that even people who had no understanding of mutations, variants, evolution, and things like that really now know this terminology and are even interested to try and ask questions about the huge amounts of publicly available data related to the pandemic, but also as an extension of that related to many other types of epidemics or outbreaks that could be occurring. And so what we did is, a couple of years ago, we worked with the virologists here at the Louisiana State University, understood how their expertise in virology and bioinformatics could be used for creating these training modules, and then continued our collaborations with researchers at UCSF, Stanford, Louisiana State University, Georgetown Medical Center, as well as other regional centers, for example, a large research consortium on malaria, to create project examples that illustrate how similar types of analytical methods could be applied to analyze data and understand these outbreaks of infectious diseases. So in this program, we use project examples related to the enterovirus EVD68, which was an outbreak that affects primarily younger children and happened recently in Europe as well as in the US. Malaria infection, again, endemic in Africa, but also in many regions of South America, I believe, and in Asia. SARS-CoV-2, which we all are continuing to observe, as well as the Ebola virus epidemic that not only started the first public and connected collection of next generation sequencing data that is publicly available, but also produced solid results, including vaccines and antiviral drugs that treated a disease that was 90% deadly before that. So as an extension of that, we also offer a focused analysis of SARS-CoV-2 as a training module that many of the research institutions that we work with can use immediately, because a big part of the sequencing and a big part of the research questions around SARS-CoV-2 are regional. And so by working with institutions like Louisiana State University, where we did many different projects on SARS-CoV-2, including wastewater sequencing, we can train students and researchers to be able to perform the same and ask questions of the publicly available data. And so on the Learn portal that you have just seen, you will see this kind of an overview that gives the basics of multiple sequence alignment, as well as the ability to ask questions about origins of SARS-CoV-2 or how the new variants are emerging and how they might affect the future of this pandemic. So at this point, I briefly want to introduce another element that I think is critical for those students that are serious about their interest in bioinformatics. And what we see time and time again is that students going through short-term training are unable to capture a lot of the aspects of bioinformatics, the application of bioinformatics, but also the ability to independently learn and continue to apply bioinformatics once those resources are unavailable. And so for that reason, we wanted to make sure that many of the same methods that are available through such training could be taken away with the researcher or the student after the program completion, and they're able to replicate and even use those in their reporting or ability to analyze data. And so I briefly want to show you a course that is designed to kind of give you a broad understanding of how this portal works, and that will be Introduction to Bioinformatics. So if you go to Introduction to Bioinformatics, bytes and molecules is what I wanted to say. Okay, so bytes and molecules. The bytes and molecules course really covers many of the aspects of what we just discussed for someone who is a beginner and has minimum or no background in bioinformatics, wants to understand the terminology that is used in such training, and wants to be prepared with some of the technical skills that are going to be needed. And one of the courses that will be available in this section is an introduction to coding. So coding is an important part of a bioinformatician's day-to-day life and activity, but it is often seen as an exclusive skill set that only bioinformaticians and engineers really need to have. But as we all know, in many aspects of research, just to produce the picture that you need, the figure, maybe some simple data analysis with some statistical properties, it is always useful to be able to know that piece of code that if you know how to run it, you know how to give it the right input, you can make it produce the right output that you want. And that is exactly the objective of some of this very basic introductory training that we offer in this section called Introduction to Bioinformatics Languages. And this one is an example using R. So here you will find some very basic things that you need to understand about data, how it is stored and how it is used in the coding environment. But we don't have to set up a whole environment on your own system to be able to run and produce even the basic plots that you might want to use in your research publications or exploratory analysis of a data set that could be very informative. So we offer here inside the actual lesson a console that you can run and get results right here. And as you can see, immediately, if I just run this piece of code, I am getting an error. And the reason is because obviously this path to the file does not work, right? And so one of the basic challenges that all of us probably faced when we started learning how to code, I can learn how to write hello world, but how do I actually make this code work for my data, right? And that's exactly where we start. So just by copying this link and replacing it right here in the console, I can run the code and I can get some results, right? And so right here, you can see that I can check the result. And now I have a successful result, right? So I need to learn about some of the syntax, just some basics of writing how to code. I need to understand the logic of how to find the right data and give it the right link. And then once I do, this console will check for me whether I'm doing the right thing. And now I can extend this to more advanced applications, like running a script to prepare a box plot, or maybe changing the way that this figure looks, right? And eventually I will be introduced to an assignment, and that assignment will be writing a complete piece of code, right? So if I know that I can complete this assignment, I'm not only following a step-by-step tutorial, but I'm in fact able to apply what I have learned And now I can copy this and use it on my system to be able to run that every time I face a result. So what kinds of activities are possible for this type of learning? One of the training that we do in terms of SARS-CoV-2 is performing multiple sequence alignment and phylogenomics of the pandemic outbreak in different regions to really understand the emerging variants or clades that have been reported throughout the course of the pandemic. So as you can see, the logic of analysis is first done on the platform. And then we actually go through a series of steps to generate a variant report. And this is a report not unlike what people use when they use clinical reporting or when that reporting is being submitted to maybe public health institutions to understand whether there is a particular variant of concern, or maybe there are some number of samples that indicate that there's a spread of that variant of concern. And so we go through preparing such a report in R, how to install all of the different tools that are needed on your own system, and then how to actually run that with the specific types of variants that you might be interested in. So it really provides an ability to apply the logic, try it on publicly available data, and potentially then apply the same to novel data that is being collected. Now, we don't stay at the sequence level. We also try to incorporate the association between sequence and the structure. So we look at the spike protein, we understand the spike protein and even map the mutations and understand the relationship between the proteins so that we understand how they are interacting, and as a result, understand the significance of some of those variants. And all of that is done in under a month-long training program. Now, another big part of what we do, going beyond the traditional bioinformatics, where we deal a lot with sequence data and visualization, is actually learn about data science. And for data science, we have this very basic introduction that I just showed you in R and Python, but we also want to be able to apply some of those methods of exploratory data analysis, statistical analysis, machine learning in a more advanced environment. And so we created this program to offer greater insights into what is possible just knowing some of those basics. Again, this is project oriented. So we start with seemingly simple challenges. For example, in the context of precision medicine, can we identify a patient that is likely to respond or not respond to a particular type of medication? And then we use that information to be able to explain some of the additional downstream requirements for dealing with bigger data, how you can use different packages, how you can produce advanced visualizations, how you can apply similar approaches to different types of data, be it genomic, metagenomic, transcriptomic, and others, and eventually how to also apply this using machine learning, advanced statistical analysis, and an understanding of how to apply these skills to bigger data sets. Now, we've recently completed a program like this that was, all of it was designed around Python, from very basics to more and more advanced types of applications, where we also highlighted the significance of an online cloud-based environment to be able to write reproducible code. And I think those are two very important trends that students need to be introduced to as they move into the application of bioinformatics or data science to particular types of research. Not only do we need to be able to extract meaningful insights, we also need to know how we got those insights and provide an environment where others can easily reproduce the same code that we write, which means that the code needs to be readable, which means that we need to explain what we're doing and why we're doing that, and also highlight the results with a piece of code that somebody can take and run and get the exact same thing. And so there we cover things like dimensionality reduction, ordination and embedding. We look at different types of classification models, including the understanding of how those models work and how principles of those logistic regression, for example, methods are used in more advanced methods like deep learning, and also connect that with, again, practical examples. And those practical examples could be used for projects that have made a significant impact. So after we cover this domain of machine learning, we actually take a look at several different publications where all of the methods that we cover are explained in the abstract itself, and the student can really see and appreciate how the specific outcome from some research really affected the industry or even the clinical use of those methods. So for example, here, modeling precision treatment of breast cancer is a project that we use to illustrate individualized and personalized treatment, as well as predictive modeling to understand potential response that does not require data from that patient themselves over a course of different types of treatments, right? Which is obviously not possible. And I think very relevant to the medical field, which is moving further and further into the adoption of such tools and the precision treatment of different types of complex diseases. Now, beyond this training, and I'll be done here in a couple of minutes, we offer to use the same elements that I've just described, the T-Bioinfo platform for no-code bioinformatics, the coding elements that you can embed right into your coursework, your expertise in projects by illustrating a specific type of a method in application to a data set that you understand and might have published before, as a resource for researchers and faculty that would like to incorporate their own training and make it available to the NAIPI network. So one of the ideas is to identify gaps. For example, if you take an institution that might not have a full bioinformatics core, and they might not have the facilities to be able to support bioinformatics and computational biology research, we would like to match them with another institution that might have those capabilities, but might be lacking another capability that that university does have. And so by linking those, we're able to address the gap across the whole network. One example that I would like to show is a course that we designed together with Dr. Sona Vasudevan. She is the program director at the Georgetown University Medical Center for a program called Systems Medicine. And her specialty is in bioinformatics using 3D protein structures and really kind of looking at the relationship between sequence and structure. And so we worked with her to create a summer course where her students and students from other backgrounds could join and learn the basics of genomics in the virtual lab. We also worked with researchers that specialize in oncology, developing a program on precision oncology. And we also kind of design these together in collaboration with other researchers and faculty. And we're hoping to expand our network and really kind of build the same model of the context, the methodology, and examples of applications using curated data sets to expand the library of resources that we're offering. Now, we've done these programs over the several years now in several different institutions. We do have a lot of kind of feedback from different users, including students, faculty, researchers, and we are starting to work with industry as well, not only to provide the basic training and the path to research, but also for students that are interested in a path to an industry career, right? Where bioinformatics is a highly desired profession and skill set and can really make a difference in someone pursuing that direction. So thank you again for listening to my part of the presentation. It took a little bit longer than I anticipated, but I would like to pass this on to Dr. Mohit Mazumdar, who will speak about some of our ongoing programs that we have started in January and are now